All right. So thank you again, everyone, for joining. It's a great pleasure to have Maria Filippetti today. I was very excited when reading her paper about data analysis with infant datasets because I guess we really need to have guidelines about how to analyze infant data. fNIRS lacks recommendation. The fNIRS field doesn't have precise recommendation, especially for infants. So it's always very nice to see someone who tackles the issue and compares different approaches and shares the results. So very, very much welcome, Maria. So about this webinar, so we are NIRx, NIRs manufacturer based in Berlin. We do various things, including some laser tag, as you can see. But more seriously, you can find us on all of these social media platforms. You can also subscribe to our newsletter here to be updated about everything that's going on in the field. Regarding this webinar precisely, you are muted. Questions are welcome at any time. Please share them in the Zoom chat if you haven't already shared them when registering to the webinar. The webinar will be updated in the upcoming days online, including our YouTube channel. And if you have questions later on after the webinar, you can also reach out to us at consulting@nirx.net or support@nirx.net. We will gladly reply to you. So who is doing the webinar today? So my name is Cecile. I am a scientific consultant at NIRx. So I was myself an fNIRS researcher before. And together with me is Camille, who is taking care of all the logistics. She's the magician in the background. So thank you very much, Camille, for making this happen. And of course, our speaker, Maria Laura Filippetti, who is a developmental cognitive neuroscientist investigating the development of body representations. Her research combines behavioral and psychophysiological methods to study the role of multisensory integration in how infants learn about their bodies. She is currently an associate professor in the Department of Psychology at the University of Essex, where she's also part of the Essex Baby Lab, of course. And yes, her research is funded by the Economic and Social Research Council, the Academy of Medical Science, and the British Academy. So congratulations, Maria, for this wonderful career, and the stage is yours.
Thank you very much. Thanks very much for the invite. I will try to share my screen now. Thanks for the lovely introduction, and thank you for giving me the opportunity to present this webinar. As already introduced, I will be talking about different analysis approaches to evaluate fNIRS infant data. This is generally a little different from the usual talks I give, because it's more focused on the methods rather than the actual research question. And so bear with me. And I'd like to mention before getting started that this was very much a team effort. It was a fantastic collaborative effort made possible by complementary expertise in computer science and machine learning from Javier Andrea Perez, and developmental neuroscientific knowledge from my colleagues at Essex Baby Lab, Karina de Klerk and Sylvia Rigato, as well as the really insightful hands-on work from Essex graduate Chloe Richmond, and of course all the participants there and that took part in the studies. As ever with developmental research, we are very grateful for the help of our families. Before getting started, I just wanted to mention that this registered report that I will present is published in NeuroImage. All scripts are available online, and I put in here also a QR code so you can easily just get to the paper by scanning the code. So during today's presentation, I will go through the outline that you can see in this slide. So first, I will provide an overview of approaches to analyze fNIRS infant data, and I will spend very briefly a couple of slides to mention why we decided to go for a registered report and what I think might be the benefits of this publishing format, especially when it comes to providing some sort of like standardizations of methods. And then I will delve into the study and finally provide some, I think, discussion points and what I think might be recommendations for developmental researchers interested in using fNIRS for their studies. So getting started with the approaches to analyze fNIRS infant data, I believe I don't have to explain to this audience what fNIRS is, but I think it's helpful to just remind ourselves some of the key points. So as you all well know, over the last 30 years, fNIRS has become a very popular tool for clinical and psychological applications. And what fNIRS does is basically recovers the hemodynamic response related to functional brain activity. And it's a technique that uses near-infrared light to detect changes in oxygenated and deoxygenated hemoglobin concentrations, which are the consequence of the increase in oxygen demand. And these are taken to indirectly reflect functional brain activity. So changes in concentration in oxy and deoxyhemoglobin during functional brain activity, brain activation can be derived through light absorption measurements. So fNIRS has many advantages, many benefits. It provides a relatively a good temporal resolution, and yes, a better spatial resolution than, let's say, EEG. It's easy to use with babies because it's relatively comfortable and allows for studies that are lab-based as well as more ecologically valid studies. It's very helpful and easily applied to a variety of different contexts. However, one of the disadvantages of fNIRS is, I believe, the lack of standardization in data analysis. And indeed, this is due to the variety of NIRS systems available, but also the heterogeneity of statistical softwares and inbuilt analysis scripts that are available across different infant labs. And all these different factors contribute to making it difficult, if not impossible, to establish the presence of consistent and reliable findings across experiments. So what are the approaches available to analyze the data? So traditionally, fNIRS data has been analyzed with averaging techniques, baseline corrected averaging techniques. And these involve averaging together the neural response of blocks belonging to a given condition and performing, as the name suggests, a baseline correction. And the processed fNIRS data is then analyzed using ANOVAs or pair sample channel by channel t-tests to compare these experimental conditions. Because of the multiple comparisons, usually what happens is that researchers, to control for false discovery rate, they apply multiple comparisons corrections. The obvious advantage of this method is that it's relatively easy to implement with NIRS infant data and avoids assumptions about the shape of the hemodynamic signal and its time course. But at the same time, the disadvantage is that they do not make use of the relatively high temporal resolution of fNIRS, which is an important factor to bear in mind. So to get around this issue, GLM methods or general linear model approaches have been used, borrowed from fMRI techniques to consider the whole time course. And so what happens with GLMs is that they provide more statistical power because of that. The GLM approach basically consists of modeling pre-specified regressors which are then convolved with the expected hemodynamic response function, so the HRF, and fitted to the data. So, as I said, there's the advantage of GLM considering the whole time course of the data, and it's relatively straightforward to apply by using ad hoc toolboxes such as the NIRs SPM toolbox. However, there is a disadvantage of the GLM that it does assume that the HRF in a given brain region is time invariant within a participant, and this might not always be the true in infants. The other issue to bear in mind is that the HRF is not well established in newborn and young infants. There are ways of getting around this a priori estimation of the HRF, which I will explain a little bit more when I go through the data analysis for the study. But this is just to give you an overview of what the GLM approach does. Now, the third approach that I want to go through is the MVPA, the Multivariate Pattern Analysis approach. So the purpose of multivariate analysis generally is to identify significant encoding of information. And this is regardless of whether the overall activation is significantly different from zero or your baseline. MVPA techniques basically allow researchers to investigate whether a pattern of fNIRS activation can discriminate between two or more conditions. The advantage of MVPAs is that it can accommodate spatial uncertainty by comparing response patterns across multiple locations at once. So with fNIRS, where anatomical registration or structural imaging might not be possible, MVPA improves sensitivity and might sidestep the challenges of anatomical alignment, especially when we're looking at different age groups as well, such as in longitudinal studies. So there are obvious advantages of using this more sophisticated approach. However, I think it's always important to bear in mind that there's no one size fits all, and that different approaches provide, as I've tried to explain through these tables, both advantages and disadvantages. One aspect to bear in mind with MVPAs is that although it might be able to provide or detect the code patterns of more subtle brain activity that might be not possible with univariate approaches, the MVPAs might sometimes not account for certain types of confounds, such as individual differences. And it has been shown in the literature with adults that this might lead sometimes to false positives. There are ways to minimize this risk again, and whether to approach this technique might really depend on the experimental design, and as we will see also, the developmental group or your sample, basically the population under investigation. Now, based on this overview, we thought, right, so comparing different approaches, comparing these three different approaches to analyze the same fNIRS data set may give insights into, the pitfalls and advantages of each method for exploring brain responses in babies. What we didn't want to do was to statistically assess similarities and differences among the three different approaches here, because as we said, any result would be specific to that population and that experimental design. However, we thought that the investigation of similarities and differences by describing what we found and how the results may converge or differ would be anyway informative for the NIRS community. So we aim to advance the statistical analysis of infant fNIRS data by illustrating really the results of these three NIRS data analysis approaches on the same data set. And we looked at the averaging approach, the GLM-based analysis, and the MVPA analysis. Now, before getting into the actual study, I just really want to explain very briefly why we decided to go for a registered report. I thought this might be helpful, not just to tell you why you should use a registered report. That's definitely not the aim of this webinar, but it's more to share my own experience on this. So if you're not familiar with these publishing format, registered reports are a way of publishing whereby the emphasis is not on the results of the study, but is on the importance of the research questions and the quality of the methodology. What basically registered reports do is to allow you to conduct peer review on your idea and your methods before you collect the data. So whereas normally you would develop an idea, design the study, finalize how you're gonna go about collecting the data, and hopefully have an idea of your analysis, and then collect data, write up and publish. With registered reports, you develop the idea, design the study, write your intro with your research hypotheses and the methods that you're going to use, and submit these for peer review. This is called the stage 1 peer review, and which means that you get to have reviewers' comments and feedback on the work that you plan to do before you actually collect your data. And once this is approved, you get an in principle approval, which allows you to then collect your data and analyze your data as you said you would do in the stage 1 peer review. Once you finalize your data analysis, you get to write up your results and discussion and submit the final report for stage 2 peer review, where hopefully the original reviewers will assess the quality of the work and ensure that you did not do anything that you didn't say you would do in the stage 1. And if you didn't, then you get to publish the report. So why I decided to go through all these steps for this study, and this was my very first experience with registered reports, and there are many reasons why I thought that this would be a suitable approach. The first thing is to really contribute to the elimination of bias against negative results. So we know that this is an issue, especially in psychological studies, and this allowed me to focus on the actual research question and the methodology, and disregard any worry about, oh, will I find something interesting? So by focusing on the actual methods and the quality and the robustness of the methods, I could really make sure that I was carrying out the best possible version of a study, given, you know, all practical considerations and limitations. This means also that I benefited from an intensive pre-study review, and that, as we know, is not always easy to deal with the peer review comments, but I really benefited from this approach, because I felt like the reviewers were working with us to get the most out of their study, and it felt very powerful in that sense. And finally, obviously, focusing on producing an accurate story, not just worry if we didn't find what we thought we would, and disregard the actual results, but actually sticking to what the data was trying to tell us. And there are many other reasons why I thought this was a good option for us, but those are the main ones that I just wanted to share as a way, given that this is more of a methodological webinar, and the registered report focused on that. So what actually did we do? So in our study, we decided to focus on examining infants' brain responses to static phase images. And specifically, we examined the phase inversion effect. This is a phenomenon whereby perception and recognition are better for faces that are presented upright than faces presented inverted. And there's plenty of EEG studies in the infant literature that have shown that faces are processed configurally from birth and throughout the first year of life. Social stimuli, like faces, have been found to also elicit the canonical fNIRs response earlier in development than non-social stimuli. And so this suggests that faces can be reliably contrasted to investigate the suitability of different approaches, analysis techniques for the use with fNIRs infant data. However, because there's only a handful of fNIRS studies on the phase inversion effect, using these phenomenon as a contrast in our study, we thought it could provide further insight into the neural basis of face processing in young infants. And in terms of brain regions of interest, we know that within the temporal lobes, the superior temporal sulcus, or STS, has been implicated in social perception in many previous studies. And so although we analyzed the whole temporal array bilaterally in our study, we targeted the STS as an area of interest. And so we included the STS in our predictions. We set very specific predictions that slightly differed for the different approaches of data analysis. But the first set of predictions involved our quality checks for all our analysis. So the averaging, GLM and MVPA. So we wanted to effectively check that the faces would, the hemodynamic response to faces would differ from baseline. And so we hypothesized, we predicted to find a significant increase in oxy and or a significant decrease in the oxy in response to upright faces compared to baseline. And we predicted that this would not be the case for inverted faces compared to baseline. And obviously this was true across all the three approaches. And then we had specific predictions for our univariate analysis, so the baseline corrected averaging and GLM. And what we hypothesized, we predicted to find a significantly greater increase in oxy and or a significant greater decrease in the oxy in response to upright compared to inverted faces over the bilateral temporal arrays. And a very similar, obviously, prediction was applied to our MVPA, just obviously changing the way in which we look at the data with this approach. And so we hypothesized to find a significant canonical discriminatory pattern of activation, which would be reflected by an increase in oxy or a decrease in the oxy in response to upright compared to inverted faces over the bilateral temporal array. So you might have noticed that I'm using the wording increase in oxy and or decrease in the oxy. So the reason why we did that is because we know that the canonical response obviously implies an increase in oxy and accompanied by a decrease in the oxy. However, we know, especially by working with infant sample, that this is not always the case. And indeed, it's not unusual to find an increase in oxy without an accompanying decrease in the oxy. But we do know that looking at both, it's important. And so we included, obviously, both oxy and deoxy with the idea that even if we found a significant increase in oxy without a concurrent decrease in deoxy, that would still represent a significant greater response. And likewise, we might be able to find a significant decrease in the oxy, which is not accompanied by a significant increase in oxy. And that would still tick our boxes for an activation. So we did a power analysis, obviously, before running the study. And this power analysis was based on the reliability of the MVPA analysis. We based the analysis on previous work using MVPA with babies that was done by Emerson and colleagues in 2017. And the power analysis suggested that we would need 23 participants to reliably carry out our MVPA analysis. What we did, based on the attrition rate that is typical within developmental cognitive neuroscience, we recruited 39 full-term 4 to 6 months old babies, and our final sample comprised 30 participants. This is because obviously we didn't analyze our data before collecting our full sample, and we ended up with more participants than we expected originally. The stimuli consisted, so this was a block design, and we had for our baseline images of vegetables, so a very non-social stimulus. And those stimuli for the test period consisted of photo images of female faces, 5 female faces with a neutral facial expressions, either in the upright or in the inverted position. The baseline lasted 10 seconds and the experimental conditions 8 seconds. These phase stimuli were selected from the NIM team, the NIM STIM phase stimulus set by Tottenham and colleagues. We used a pseudo randomized order to ensure that a given condition was not presented more than a certain number of times in a row. I think it was 3 times in a row. And these also helped to reduce habituation effects. And we carried on with the stimuli presentations until the baby became fussy or bored, or until we reached 30 trials. We used the NIRx Scout machine to record the data, and we relied on external surface anatomical landmark to identify location of responses. Our source detector array comprised of 8 by 8 ST, and that basically made up 20 channels over the bilateral temporal array. We had a very specific set of data inclusion criteria that obviously we had to really think about carefully before collecting the data as part of the registered report. And I just want to show you what the criteria were and how many participants we exclude as a result of that. So we did not exclude any baby as a result of refusing to wear the NIRs cap, which is not as uncommon as you might think if you've not done studies with babies. The other one is showing signs of distress, so fussing or crying, parental interference, again, not as uncommon, but hopefully, yeah, we didn't exclude any participant as a result of that. The other one was signal quality problems, such as pulling the NIRs headgear or movement or poor contact with the scalp, and we excluded 1 participant because of these. None of the participants were excluded because of experimental errors, and the majority of the babies that we had to exclude for this study was because of looking time. So obviously with infant studies, we can't force the baby to look at the screen, so the babies behave as they would normally do. Sometimes they look at the stimuli, sometimes they don't, so we need to carefully code the infant's looking behavior to ensure that trials were only included in the analysis if the infants had actually watched a significant amount of trials. The criteria for us was at least 60% of the time in each trial, as well as 30% of the pre and post stimuli baseline. Okay, so the pre-processing and data analysis. We followed a very specific set of pipeline steps for pre-processing the data, and we used HOMA2 to pre-process the data. The criteria for including the data in our final analysis was to have at least 6 trials to carry out the averaging and the GLM analysis, so 3 trials per condition, and at least 16 trials to carry out the MVPA analysis. Now, we did actually change something as a result of the COVID pandemic, because this study was done during that time. So our data collections was significantly affected by that. And due to recruitment challenges during that time, obviously we couldn't collect the amount of participants needed to have at least 16 trials per condition for the MVPA analysis. So with the 30 participants that we had, we ran the MVPA with that full sample size. So those had a minimum of 3 good trials per condition. The majority of them had at least 6. And then with the 13 participants that had at least 8 good trials per condition. And as I said, we carry out analysis on the whole array. Now, to really go through the pre-processing steps, what we did was that first, the light intensity data was converted to optical density, and we used the imprune channels functions in Homer to reject channels with raw intensity smaller than a certain voltage. We then manually excluded the trials, as I said, in which the infants were not attending the stimuli, and we corrected motion artifacts using wavelet analysis as recommended by Brigadoy and colleagues in their 2014 paper. We then run bandpass filtering to attenuate slow drift and high frequency noise. And finally, the data were converted to relative concentration of oxygenated and deoxygenated hemoglobin using the modified Beer-Lambert Hall. So how did we analyze the data? For the baseline corrected averaging approach, we computed relative changes in oxy and deoxy for 18 seconds long epochs, starting 3 seconds before the onset of the stimulus and ending 7 seconds after the trial offset. The mean oxy and deoxy concentrations during the 3 seconds baseline was subtracted from the concentration in the then 15 seconds analysis period. And the signal then was averaged across channels for each channel and condition. Following previous literature, what we did was to basically quantify the mean hemodynamic concentration changes during 5 3 seconds long sub-epochs following trial onset. And then we performed repeated measures analysis on these sub-epochs within the 2 conditions, so the upright and the inverted phase. And what we did in this case was basically identify whether there was a mean effect of time when we consider both conditions together compared to baseline. And after that, we then looked at a difference in hemodynamic response between the 2 conditions, so contrasting upright versus inverted. And this would be a main effect of condition, obviously. And we did multiple comparisons corrections following the false discovery rate approach. For the GLM, we combined custom-made MATLAB scripts that are available, as I said, online, and the SPM NIRs toolbox to carry out this analysis. For each infant, we constructed a design matrix with 3 regressors. So the first regressors modeled the upright phase trials, the second modeled the inverted phase trials, and the third regressor modeled the baseline trials. These regressors were then convolved with the standard canonical hemodynamic response function to make the design matrix. And this was then fitted to the data using the general linear model as shown in the NIRs SPM toolbox. For each regressor, for each infant, we basically obtained beta parameters that were used to calculate the contrast between conditions of interest. And again, we corrected for multiple comparisons using the FDR correction with the Benjamin Hochberg procedure. For the MVPA, data analysis was conducted using custom in-house scripts, again, available online. And the multivariate decoding accuracy was estimated as a basis of the prediction of a support vector machine, which is basically a supervised learning model that was trained with specific target labels for each vector. So we had upright versus baseline, inverted versus baseline, and upright versus inverted. We used as a feature for each condition the GLM beta coefficients. So using the output of the GLM with one regressor per condition and channel. To validate the generalization of the MVPA results, we generated testing and training sets using a repeated 5-fold cross-validation process with basically between subjects, so non-overlapping subjects. And more complex steps were then taken to ensure model sensitivity, but for the sake of time, not going to go over all of these. And you can obviously check the paper if you're interested in more fine-grained, detailed. And we applied a Monte Carlo guided permutation to then perform our group level analysis. So the results. I'm going to go through the pre-registered analysis first, and to ensure clarity, I will display a table with a little summary of what we find, because it's quite a lot to take in. But the results first for the baseline corrected averaging. The initial analysis identified 3 channels that showed a significant hemodynamic response during the trial period as compared to the baseline period. Remember that we needed to check first for a main effect of time. So we wanted to compare the hemodynamic response to the baseline period when both conditions are considered together. So upright and inverted faces together against baseline. So these channels showed a significant increase in oxyhemoglobin between the baseline period and the trial period, and were channel 8, 18, and 20, as you can see from this image. Channel 8 also showed a significant decrease in the oxyhemoglobin between baseline and trial. A main effect of condition, so an increase in response to the upright face compared to the inverted face condition, indicating a greater oxy response to upright, was displayed for channel 18. So this was the only channel that showed both a main effect of time and a main effect of condition. However, when we corrected for multiple comparisons, this activation did not survive the correction. Now, GLM analysis, again, these are the pre-registered analyses. The t-tests on the beta for the contrast between the individual conditions and baseline showed no significant channels. So no significant increase in oxy or no significant decrease in deoxy in response to upright face compared to baseline, or in response to inverted face compared to baseline. So our GLM output data did not pass the initial quality check here. When we look at the t-tests on the beta for the contrast between upright and inverted conditions, we found 2 channels that were sensitive to phase orientations. And these were channel 8 and channel 1, as you can see from this image. I will go back to what's happened here with our exploratory analysis. So unsurprisingly, given that the GLM did not pass our quality check, the decoding accuracy of the MVPA, which was based on the beta coefficient from the GLM, was not significant for any of the comparisons. So we ran the MVPA with both the full sample of 30 participants with a minimum of 3 trials per condition and with the 13 participants that had at least 8 trial preconditions. And for both, although the coding accuracy was above 50%, this was not significant for any of the comparisons or the chromophores. So once we ran our pre-registered analysis and we saw the data, we decided to run more data-driven analyses that were also informed by the success or lack of the approaches that we used. One of the requirements of the pre-registered univariate analysis was for the channels to display a main effect of time. And this is shown by a significant hemodynamic response during the trial period compared to the baseline period when both conditions are considered together. So the reason why we set this prerequisite was to reduce the number of comparisons. However, it is likely that some channels were sensitive to phase orientations, despite not showing evidence of a significant hemodynamic response when both conditions were considered together. And so we also report the results on main effect of conditions, even for those channels that did not show a main effect of time, if that makes sense. So these results showed that channels 9, 11, and 17, as you can see from this image and from these plots, showed a significant main effect of condition, evidenced by a significant increase in oxyhemoglobin for upright compared to inverted. And as you've seen earlier, in the baseline corrected analysis, channel 18 was the only channel that displayed both a main effect of time and a main effect of condition. Now, back to the GLM. The GLM-based univariate analysis, as I showed earlier, did not find evidence for a significant hemodynamic response between the individual experiment conditions and baseline. But I mentioned at the beginning when we were talking about the different approaches that the HRF is not well established in young babies. So it is possible that the estimation of beta coefficients from our approach that provided an a priori estimation of the HRF lacked sensitivity. So we decided to run an exploratory GLM analysis using time and dispersion derivatives when modeling the hemodynamic response. What we do with this approach is to take into account the variability in the onset time and shape of the hemodynamic response that are common in infant data. So by using this approach, the t-tests on the beta for the contrast between individual conditions and baselines showed a significant increase in oxyhemoglobin in response to baseline compared to upright phases in channel 12. And there was also a significant increase in oxyhemoglobin in response to baseline compared to inverted phases in channels 12, but also in channel 14. We then also found that channel 5 showed a significant decrease in the oxy for the upright compared to baseline condition, and for the inverted compared to baseline. Additionally, channel 1 also showed a significant decrease in the oxy for baseline compared to inverted phases. And then we did the t-tests on the beta for the contrast between upright and inverted. So we contrasted the two conditions of interest, and we found that channels 8 and 17 were sensitive to phase orientations. Again, all these contrasts did not survive multiple comparison corrections, and there's reason why we think that might be the case, given how conservative these approaches are. But for the sake of clarity, we are reporting all of these, and happy to discuss this further at the end. These are the only, we found significant improvement in how the GLM with derivatives could actually provide some significant activation that might be accounted for the fact that we took into account the onset time of the response and the shape of the hemodynamic response with this approach. So based on these results, we then finally did exploratory analysis on the MVPA as well. So if our MVPA was affected by the estimation of the betas, then we should observe that if we carry out a MVPA using the baseline corrected responses, we would show results in line with our baseline corrected univariate approach as well. So that's the first exploratory analysis that we did with our MVPA. And we use a similar approach to the beta-based approach that we pre-registered, with the exception that in this model, our features were the multi-channel vector of baseline corrected averages for each condition. And this was over time window that went from 0 to 12 seconds. We ran this MVPA with the full sample of 30 participants, and we found that the group model for oxyhemoglobin in the comparison upright versus inverted yielded a decoding accuracy above 70%, 72% to be precise. And this was significant for channel 17. This was the channel that had the higher decoding relevancy, basically. Decoding accuracy was also statistically significant for deoxyhemoglobin, and the model showed that channel 11 had the highest decoding relevancy. So these results show basically the expected significant canonical discriminatory patterns of activation in line with our baseline corrected averaging approach, suggesting that the MVPA kind of like replicated our results. But then we also wanted to examine whether the coding accuracy would benefit from the betas derived from a GLM approach that uses time and dispersion derivatives. And so we run an additional exploratory MVPA with the data set of 30 participants. What we did here was to use a recursive feature elimination to select the best channels. So how these methods works is basically we start with a base model that includes all the channels beta coefficient, and then we recursively prunes features and check for the improvement of the decoding accuracy as we remove these features. And the steps that yields the highest score is considered the best number of features. We then do a permutation test to estimate the p-values of the coding accuracy and the feature importance, so the coefficients basically. Using these methods, we found that the coding accuracy was statistically significant for oxyhemoglobin during the trial period compared to the baseline period. The decoding accuracy was around 70% for upright versus baseline, and just below 70% for inverted versus baseline. Similarly, we found statistical significant decoding accuracy for deoxyhemoglobin. When we then looked at the comparison upright versus inverted, we found an 80% accuracy of the coding for oxyhemoglobin and a 72% accuracy for deoxy. The channels that yielded the higher accuracy were widespread temporal lobe channels located over channel 5, 15, 20, 13, and 19. I should probably just read. These demonstrated a significant increase in oxyhemoglobin. And for the oxyhemoglobin, the channels that yielded a higher accuracy was channel 16, as you can see. So the results of these MVPA using beta coefficients from the GLM with the time and dispersion derivatives show significant discriminatory results in response to upright compared to inverted phases over the right temporal array predominantly. So to conclude, there's obviously a lot of channels that I mentioned and activations and analysis, but I think the take-home message is that For the baseline corrected averaging approach, we find that only channel 18 realistically it showed a significantly greater oxy response in the upright compared to the inverted phase condition, while also showing a main effect of time. Channel 18 is approximately overlying the right posterior STS region based on previous standardized scalp surface fNIRs maps. And these results are in line with previous studies using the phase inversion effect in babies, showing that channels overlying the STS regions are sensitive to phase orientation. Given that we did not find evidence for a significant hemodynamic response to the upright condition compared to baseline, we have reasons to question the sensitivity of our original pre-registered GLM analysis. And because the GLM methods makes assumptions about the shape and the timing of the HRF, and given that this is not well established in young infants, assumptions about the HRF may be susceptible to model misspecification. And so we use as a potential solution the possibility of using a GLM with time and dispersion derivatives, which takes into account the variability in the time and the shape of the hemodynamic response. And we found that this analysis was potentially more suitable for our investigation. And unsurprisingly, given this potential issue with the GLM approach, the MVPA analysis that used the beta coefficients was not able to distinguish between the two stimulus conditions in our fNIRs data with what we thought was our anticipated level of accuracy. Therefore, we ran exploratory analysis that demonstrated a higher decoding accuracy based on baseline averaging, and the MVPA that was based on the beta coefficients derived from the GLM with the derivatives. These approaches yielded a much higher level of decoding accuracy for the phase orientation contrast, and we showed that a large set of channels lying over the right STS region of the infant brain contributed to this higher decoding accuracy. So based on what we did, we put together a small set of what we think could be recommendations or considerations for future studies in developmental research. The GLM approach that we carried out might have not been suited for the analysis of the current data set, but however, we think the GLM does provide clear benefits for NIRS studies. For example, Pinty and colleagues have shown how GLM approach is particularly suited in cases of covariates, so where we are considering additional regressions such as other physiological measures or behavioral responses. But obviously, with younger babies, I think it would be really important to consider GLM approaches that take into account the HRF variability. Obviously, one approach is the one we used. However, there are other methods that can extract hemodynamic functions from fNIRs, such as the finite impulse response filter approach. More refined MVPA approaches might also be helpful in modeling the uncertainty of the input data. And so we recommend using beta-based MVPA approaches with experimental designs and populations that are conducive to eliciting a clear canonical hemodynamic response. Otherwise, there are data-driven approaches that might be particularly useful in modeling the uncertainty of the data, such as MVPA that are based on explainable artificial intelligence. Overall, I think my future take is that it is important to consider the use of different analysis approaches to look and examine the results of your data to really provide, assess, let's say, how robust one's conclusions are. And I think this was a really great learning curve for us to, and a first stepping stone to establish more robust and standardized ways of analyzing fNIRS data in babies. So thank you very much, and I would be happy to answer questions.
you so much, Maria, and for this very in-depth talk. We received a few questions from people, from attendees when they registered. So unfortunately, they were not signed, so I'm just going to read them and let you answer them. Thank you so much for those who wrote them. So first question, would it be possible to discuss functional connectivity analysis with infants? I would say this, we have resources on our website. Since it's not the topic of the webinar, I would let the person who asked this question would invite you to check our website, YouTube channel, and resources. And if necessary, write to support@NIRx.net and we can guide you through resources. Second question, what software is being used? Okay, so this is during the webinar for analysis. So Maria, you replied that was Homer 2 and NIRs SPM, if I remember correctly. Okay. Please share resources on data processing and major papers in this field. Again, this I would refer to our support website. We have plenty of resources, YouTube channel as well with previous webinars. Question 4, could a brief on which analysis method to adopt depending on whether the paradigm, yes, this Maria, your input would be very valuable, I think. Which analysis method would you recommend depending on the design is block or event related? Do you have any recommendations for this?
So actually, originally, we really wanted to contrast event-related and block designs as well in our study. However, it was a bit too much to put everything in there, as the reviewers also suggested. So I don't have a clear response, purely because I think there's been studies that have used both approaches, both designs and different ways of analyzing the data. I don't think there's an overall agreement of there. I think there are many examples of regression-based event-related designs in fNIRs with adults that used GLM-based time series regressions. I'm thinking of early 2000 studies and GLM-based analyses are able to temporally estimate temporally overlapping events. So I think GLM would be suitable in event-related studies, particularly. But I don't think there's been any studies that have actually contrasted different methods of analysis using these different designs. So that's probably something that I would welcome.
Yes, I don't have any study popping in my mind either. Maybe Armin, you have some ideas, but yeah, we can discuss this later. Thank you very much. So question number 5, what are the things we will have to follow for analysis? Yes, this is a very general question. Please go to our help center and we can guide you through resources. How do you deal with non-stationary responses such as habituating responses?
I think this is a general concern in studies. So I suppose this means like repetition effects, right?
I think so as well.
Yeah, so obviously repetition can influence the hemodynamic response by, and we can see either an enhancement or a suppression. In babies, we sometimes manage to get around this issue by carefully designing the experiment with pseudo randomization. So that's one of the things that we did in the study to ensure that the same stimuli or conditions would not happen, would not be repeated within, let's say, 3 repetitions. I think generally, one thing that's been done in the past with the more classic averaging approaches was to contrast the hemodynamic response in the first half of the experiment against the second half to see whether there were differences in the hemodynamic response. And that's something that I know has been done to carefully check for potential habituation. So there's always ways to minimize the risk, I think, at the design level. But if one is concerned, there's ways in which you can look at changes in the hemodynamic response. With GLM, I think, particularly with babies, applying these methods that take into account the variability of the onset and the shape are particularly beneficial to address these issues as well.
Mm-hmm. Thank you. Sorry, I have to go very quickly. That every question would invite an in-depth discussion, but for the sake of time, let's address the other questions. Okay. Yes, so the rest are general questions. So I would invite people to look at our support center now about the chat. So Maria Piedarita, I'm sorry if I pronounce your name wrong, is asking, in the GLM approach, do you take into account the HBR signal convolving the design metric with an HRF with the shape of the HBR? Or do you just take into account the canonical HRF with the shape of the expected response of the HBU? Also, how is the shape of the HRF that you use?
Okay, I think it's the latter, but I would have to double check because on the top of my head, I can't remember exactly what we did in that case, but it's probably in the paper. Sorry, I don't want to give the wrong answer, so I'm going to say probably the latter.
Okay, so maybe this person can check with the corresponding author on the paper and write to the corresponding author.
Yeah.
Okay, now Dorde is asking, would you say that the bad beta coefficients from the GLM affected the MVPA more than the fact that you had to use minimum 3 good trials instead of minimum 8 good trials like you had initially wanted? I'm not sure the question is clear. Dorde, if you're here and you want to open your mic and ask the question directly to clarify, maybe. Yes, no.
I can guess. I can try and guess.
I'm sorry. Can you hear me okay?
Yes.
Just a second. So my question is essentially because you had mentioned that because of COVID, you had to change the minimum amount of good trials that you used for the multivariate analysis method. And I was wondering if you think that that had an effect of how it turned out in the end, rather than just the fact that you had the bad beta coefficients that you used for the features.
So we run the analysis with both, and I think the results aligned. So I think it was, my feeling is that actually in the GLM that we originally used affected the results of the MPA more than just having an underpowered sample or having less trials than anticipated, because the results were going in the same directions for both data sets. But yeah, ideally, good. I think they also had follow-up questions.
Yes, is the workflow at your lab when doing infant research to use all 3 of this analysis method? Then what is the biggest issue you have found so far in analyzing infant data? And what would you say one would have to watch out for most?
Right. So no, we're not analyzing all our data using the 3 approaches. I think the take-home message is more that depending on the experimental design and the population, you can choose which of these approaches might align best. I wouldn't necessarily exclude running more than one type of analysis if you think that's going to be valuable for the research questions that you have, but that's not necessarily my recommendation to, try everything. The the biggest issue. I mean, with infant data, I think there are many issues generally to deal with. I don't think there's one that strikes as being the biggest. I think it's more about carefully thinking about how the experimental design and the analysis pipeline would be the best way in which you can make sense of your data. Infant data can be particularly noisy, despite fNIRS being, quite good with motion. So ensuring that the way in which you carry out the study provides the best possible way for you to get meaningful data and reduce attrition rate. So I would really focus more on the implementation, so the data collection, and making sure that you have everything in place to make the most out of your sample, and then think about what to do with it in the data analysis process, if that makes sense.
Thank you very much. So last question from our colleague Armin. Given the clear issue of the HRF variability, what do you think about the recent idea to use a trial-specific HRF from Prince et al. 2022 publication? This approach has been successful in fMRI research. I have never heard about this, so I'm very curious now.
So I have heard about it, and I recall talking about this with somebody, but I don't know if I have an opinion because I've not read the paper. So I will read the paper and hopefully will get a clear idea. I think in principle it could work well, because I suppose the way in which it works is that you create your HRF based on individual trials, and so it takes into account variability in that sense. Is that?
Yeah, if I may answer, it basically uses a predefined database of HRFs. I think there's a couple of databases. Of course, we can discuss if they work the same way for grown-ups and children in the same way, but I think they have like 20 basis models, and you run a single trial GLM. But I think the kicker for this method is that you calculate an inter-trial reliability index between the beta values. And based on this, and also a specific denoising method, you can basically find the optimal response for every trial and voxel, because it's based on fMRI data. So it's quite complex, but there's a nice Python workflow that one can use. So fMRI data, and I mean, if you want to save you from reading the paper, there's also a nice YouTube presentation by Prince that explains the methods quite nicely. So I would anyway like to test it myself for fNIRS data. But anyway, thanks for a nice presentation, and I'm really happy to see somebody looking for standardization of methods and comparison. It's highly appreciated. So thank you.
Thank you. I appreciate that.
So on this, I think it's time to close this webinar. I'm very, very happy. It looks like there is plenty of discussions to follow up with. So please feel free to reach out to us and the corresponding author on the paper. Thank you everyone for joining, and see you in the next webinar. Bye-bye.